First, I'd like to talk to you, or give you a general overview of the topics I'm going to be talking to you about today. The first topic I'm going to talk about is just a summary of the study that was performed. The second is a little bit of a background of ctenophore biology, which you can see here. If you're unaware of what a ctenophore is, it's a good image here, kind of alien-looking figure. And then next, I'll talk to you about the 5 clades and the different various hypotheses of cladograms that are created from the phylogenetic relationships between these organisms. And then I'm also going to take you through the maximum likelihood analysis that was conducted in this experiment, along with postsynaptic and myogenic gene presence. And then I'm going to finish with a brief conclusion. Now the study was performed to see if ctenophores were in fact the oldest sister lineage to the rest of the animal kingdom of multicellular animals. The main species used in this species was the ctenophore Nemniopsis liddi, which you can see right here. And previous to this, the main hypothesis was that sponges, or porifera, were the oldest sister lineage to the rest of metazoa. But this one is challenging it. And these share a complex cell types with the bilaterians, which is the clade that we belong to. And it goes through, it's determined through phylogenetic relationships, and it yields to new discoveries. Now, ctenophores, they're also known as comb jellies, or sea walnuts. This comes from the way they look, so it almost looks like a walnut. And the combs, or the rows right here, looks like a comb type thing. So that's where it gets its name from, comb jellies. The adults grow up to approximate 10 centimeters in length, and their comb plates, they have comb plates in rows. They consist of 8 comb plates, ciliated comb plates. And this picture here is the sidipid stage. So here you can see the tentacles that come out from it. These can be contracted back in through different muscle cell types. Now, they're not jellyfish. A lot of people think that because they look like jellyfish that they belong to the same clade as jellyfish, but they don't. The biology is critical for reconstructing animal evolution, and until this point, there's never been a full genome that's been sequenced that belongs to the ctenophore clade. Now here is just a brief overview of the cell division that goes on through the embryo of ctenophores, specifically Nemniopsis liddi. Figure D here is a fertilized egg or an embryo, and then E through F is the cleavage stages that it goes through to create a blastula. Now, figure I down here is the gastrula stage when the blastula collapses, and it begins to form later development of the N liddi, which is the different cell types. And then it comes around and grows into this. Now, the 5 clades of animals, it consists of the porifera, the placozoa, the ctenophores, the cnidarians, and the bilaterians. Now, the first hypothesis that I'm going to talk about is in figure A, you can see here, and it's the Coleanterata hypothesis. This groups cnidarians into a group or a sister lineage with the cnidarians. This is because of their physical makeup. It gives it's grouped into a body shape with a teacup shape at the top, one opening, and then the opening is lined with tentacles. Now the second hypothesis, it puts its sister to bilateria. This is based off some linkages between mesodermal genes that are present in both. The third is figure C here, and it puts sister to porifera. This is up till before the study, this was the most common hypothesis of the animal lineage. Figure D is the one that we're looking into, and it puts ctenophore sister to the rest of the metazoa. Now figure E is the placozoa sister group to all metazoans. This is based off its physical makeup. Placozoas basically are one species. They're debatable several species in the placozoa, but they're very basic forms. So this is based off the way they look and their advancements in their body shape. Now the fourth, or figure F, puts ctenophore or groups all diploblastic organisms into one clade in comparison with bilaterians. Diploblastic, they have two embryo layers, and then the bilaterians have 3. Now this is a bit more of the study. This study is very sensitive to the types of gene selection, the taxon sampling, and also the model selection used. It makes the understanding of animal evolution difficult because of this sensitivity. DNA was isolated from 2 self-fertilizing embryos from 2 different darians, ctenophores, and 2 different sequencing instruments were used with maximum likelihood, sequencing the gene. And MLD was among the small 7% of genome sizes in the multicellular organisms, and it's very densely packed with low repetition. So this means that when they're sequencing the gene, there's not a lot of introns, there's not a lot of wasted nucleotide pairings. So a lot of it is very good information, and it's done very quickly. Now here is the maximum this is the various hypotheses put into, basically split into 2 different groups. It analyzes genome sets, full genome sets of 13 different animals, and then it also splits it into EST sets, which are smaller, more specific DNA sets. And this was about 58 animals compared within this grouping. Because there are it's not a full genome, so it's a lot of different mRNA, tRNA, cRNA. Now these 2 categories are split into another 2 categories, one using the maximum likelihood method to analyze them, and then the next is using the Bayesian analysis, and it uses the CAT model, which is based off topology, so the shape of the map, the branch lengths within the map, so the length of these branches connecting, and also time reversible, so it puts these species into different spots within the dendrogram. Now these 4 clades here that you can see right here are outgroups. They're not part of the unicellular organisms. They're thrown in there to see what effects they would have on the data, and if they would create a different topology. Now the maximum likelihood, it showed that the most likely outcome, the ctenophore is sister to the rest of the metazoa. And the outgroups apparently had no effect on this. Now, the Bayesian analysis, on the other hand, it grouped peripera, as you can see right here, peripera is sister group to all other lineage. So it said that that was the most distant lineage in the multicellular organisms. Now this was, you can see above here, there's some decently sized numbers in here, so the higher the number, the more likely it is that this is the hypothesis. So these numbers right here, they would believe to have resulted because of poor taxon sampling. So it's the lack of full genome sets used. Now, this in the gray box right here, the reason why it's in a gray box is because this data was insufficient. It had a hard time converging with one another. These numbers are based off of very small convergence. So it didn't, because of the vast amount of animals used in this, it had a hard time converging. Now this here in the light shaded blue box, that's outlined in red, this was done to basically take out the outgroups, so these groups right here. They wanted to see what would happen if we took out these other effects, because people have how they believe Nadarians were related to ctenophores. They believe that these outgroups were having an effect or masking that relationship, so they decided to take out the outgroups to see what the results would have been if the outgroups weren't involved. And it still put ctenophores as a sister group to all other lineages. The conflict between the Bayesian analysis and the maximum likelihood leaves room for debate. So these numbers here and these numbers here, it still provides opportunity for this hypothesis to be questioned. Now, a weighted likelihood was taken from this data, and this is what resulted from the maximum likelihood analysis of the ESTs, which is the smaller sampling sizes with the 58 different species. Now, don't be overwhelmed by all of this in here. What you want to focus on is the outside here. And it also, you might recognize it, it looks like figure D, which is the hypothesis that we were trying to prove at the beginning of the study. Now, yeah, it puts ctenophores, sister group to all other lineages, and then it has the other groups before that, which is the unicellular organisms. Now, this is the maximum likelihood results of the gene content. So this is the full genome sequences, so you can see there's a lot less species involved, and it has the same outcome. So it has the same, it puts ctenophore as the sister group to all the lineages. Now here, this looks more at the transcriptomes. So this is tRNA, RNA, cRNA. And this here is a figure of postsynaptic genes. So this is receptor genes that are present in all of these organisms being compared to all these clades. Now what we're looking at here, all the blue is what are present genes in all of the clades. And the underlined means that they're present in Amylidae, which is the ctenophore being analyzed. And what's interesting about this is that periphery, which is in red, you can see here, so it has all the blue colors and then it has these additional red. Periphery doesn't actually have a nervous system. They have the genes to provide for a nervous system. And so this provides an interesting outcome, seeing that it actually has additional genes in comparison to the ctenophores. Now right here is the myelogenetic components of the Amylidae. This is being compared to bilaterians. Right here in figure A, this is a figure of the smooth muscle genes that are present within cnidarians or ctenophores and bilaterians. And what you'll notice here is all the blue are genes that are present within ctenophores. And the red here, which is troponin, gene for troponin, is not present in ctenophores, but it's present in bilaterians. And this gene is a gene that's used to produce more smooth muscle. Now this figure right here is a comparison between the mesodermal germ layer that's produced in the embryo of the genes that are involved in creating that. Now the Amylidae does have some of these genes which are in blue, and the bilaterian includes the blue and the red. So this provides a question of, okay, none of these other clades have these genes to produce a mesodermal layer, but ctenophores do. So it questions the accuracy of this hypothesis because it believes that these must have been very close relationships to bilaterians. But this also could have happened after it diverged from the different clades. So it could have happened early on. After it split up, it could have developed these different genes later on. Now to conclude, the evidence supports a new view at the evolution of cell types throughout our history. Just to cover it again, this is the organism that was used within the experiment, and this was the resulting hypothesis, putting ctenophores sister group to the rest of the metazoas. But this, however, again, is very questionable. It's not 100%. This data is not 100% in concrete, so there's still a lot of questions to be asked about it, but it provides a lot of steps towards future analysis, because this is the first time that ctenophores' full genome has been sequenced. So it provides a lot of opportunity to further insight into the animal lineage and cell type development throughout evolution. Now, acknowledgements, I'd like to thank Dr. Clements for all her help, and Dr. Vaximanis. He was a scientist that was involved within the research. He's very helpful, surprisingly helpful, so I want to take a moment to thank him as well. And it's just